Starchild was found in Mexico about 1930 by a young girl. She doesn't know how old she was, 12, 13, 14, 100 miles southwest of Chihuahua, which is there, and that's in the range of the Copper Canyon area. It's high desert area. Next slide. High desert like you would find around the Grand Canyon.
Just
Get a hammer.
Press. That's it.
High desert area like you'd find around the Grand Canyon or something like that. And in a cave, in a mine tunnel in the area, in an abandoned mine tunnel, she was out exploring, as kids will do, and she had a basket with her, and she found up in the mine tunnel, where she could still see, lying on the floor of the mine tunnel, was a skeleton, a human skeleton lying on its back. 1930, a kid didn't freak out when they saw a skeleton, dogs and things in that area, because the bones would stay. So she noticed beside it, when she got a little closer, was a mound of dirt, and coming out of that dirt was what she called a misshapen hand. And that misshapen hand was wrapped around the upper arm bone of the skeleton lying on its back on the dirt. That one's in the dirt, but the hand's coming out. So she knows something's in there, and she can see, because it's in a mine tunnel, it's never been compacted by rain or anything like that. So with her hand, she's able to dig it out, and she exposes the whole skeleton. She tries to recover the two skeletons. It's a long story. She tries to hide them. A flash flood washes everything away, and all she's able to recover is the two skulls with a little bit of damage to each from being tumbled down in the flash flood. Long story, not worth getting into it here. So she recovers the skulls, and she brings them home, and she keeps them as souvenirs. And as she grows up, she shellacks the skulls to preserve them, which people did back then. Puts them in a cardboard box, keeps them her whole life, basically in her garage in El Paso, Texas. And as she's dying in the early 90s, she wills them like you would pets or something like that to some friends. Her husband was a border patrol agent. He wasn't that comfortable with having two skulls in a box in the garage. So the friends say, yeah, we'll take them, and they keep them for 5 years, but he's an FBI agent. He's not that comfortable with it either. So then they meet this couple called Ray and Melanie Young, and Melanie is a neonatal nurse, been a neonatal nurse for 15 years. So he said, would you mind having a deformed, because that's what they believed, that the weird skull was a deformed skull. Would you mind having a deformed skull? And, this lady died, and we don't want to throw it away. And they said, yeah, sure, we'll take it. So next slide. Ray and Melanie, again, all these people in El Paso, Texas, Melanie, Ray, obviously. And Melanie had been a neonatal nurse, as I said, and she took one look at it, and she says, it's not a human deformity. That is too light, it's too symmetrical, it doesn't look anything like a human, it doesn't have human anything. This is not a human deformity. And both she and Ray were members of MUFON, Mutual UFO Network of El Paso. And so Melanie says to Ray, I think it's the skull that would fit in the head of a gray alien. Because when you hear the prototypical descriptions of gray aliens, they have that large kind of heart-shaped build here with that crease down the middle, and they have that very fine pointed lower face, and she says it's got all of that. I think it's that. And he says, okay, well, we'll find out, but he's not as convinced as she is. So they take it out to a couple of experts in El Paso, real true experts from the universities and stuff, and they just tell them immediately, it's a cradle-boarded hydrocephalic, take it home, forget about it, don't worry about it, it's nothing, it's a cradle-boarded hydrocephalic, which I also heard many times. It's the standard reply of somebody who doesn't want to deal with it, who doesn't know what they're talking about, and they just want it out of their place, out of their space. So when they got that, Melanie says these people give us a run around, we need somebody that'll work on it. So they went to the El Paso MUFON meeting, and they said, who in the field of alternative knowledge knows anything about skulls? And that's how my name came up. They said, there's this guy in Louisiana, Lloyd Pye, he talks about skulls. In my other work, with hominoids and human origins and things like that. So everybody always asks, how'd you get it? That's how I got it. So they said, the next time you're in El Paso, would you come by? We have something to show you. They showed me, and I thought it had to be some kind of deformity, but I didn't know what. I'd never seen anything like it before, but the idea of holding an alien skull in my hands was at first very impossible, just couldn't be. like finding the Dead Sea Scrolls or something. Couldn't do it. But by the end of that year, but I said, it is very weird, and I will try to find out for you what it is. And I began to take it to experts, and I began to get the answers that I would get from the expert, whether it was ear or brain or neck or face or head or whatever. I can't explain my part of it. I've never seen it, but I know it has to be some kind of deformity. Everything else has to be covered, but I can't explain my part of it. We have 10 or 12 guys say, I can't tell you my specialty area, I can't explain it, but it has to be some kind of deformity. You know they're all just flailing for an answer. Anyway, next slide. This is the two skulls lined up together. That's the human one that was found. A couple of different shots here. And that's, of course, the Starchild. And you see the differences in every position. You have 25 major physical differences. There is not a single similarity on this skull to that skull. There's not a point of reference on the skull where you can say that's exactly the same in human and Starchild. Everything is different. Next slide. We start out here with the front view of the adult female and the Starchild skull. Note the eye sockets, the difference in the eye sockets, the brow ridges, you know, this swelling here is a brow ridges, nasal dip, you know, a dip in the nose, no dip in the nose. Completely different starting out at those basic things. Next slide. Profile, you see that a human eye socket is about 2 inches deep. My finger would go in to that much. If here, the Starchild, it would go like about that. It's about a ½ inch. So you have an amazing difference there. Then you have, again, the brow ridges. All humans have that. The Starchild doesn't have a trace of brow ridge. You have the dip in the nose where you come down off the brow ridge to the nose. Starchild goes, as you see, just like a ski jump right down. So completely different, just starting again at these very basic things. Next slide. With the profile shot, you hear these brace bones that you see here. People say, what's this? It's called a pterygoid plate, and you have two of them, one on each side of your face, up under there, and it braces your maxilla, right? So if you get a really good shot in the grill, it doesn't just go right back in and crush your brain and kill you. So this is one of the most important bones in your body in terms of physical impact in your face. Got to have a pterygoid plate, you're no good. But as you see, the size of this one versus the size of this one. The ear holes are different too. This is shoved over, but that does have an ear hole. We'll talk about this, but this is the zygomatic arch, and you see here it flares out and goes around. Here it's tucked in much tighter to the face because it's going to have a much tighter face. And the inion, we all have an inion. Anything with a head on a neck has a spot back there. You can all feel your own. You have a knot back there. It's called the inion. It's where your neck muscles attach. You see the human has one, Starchild does not. In fact, it has a dent, you're going to see. It's not just different, it's like super different. Next slide. Okay, top view crowns. You see that a normal human skull is rounded in the crown back, and the Starchild has an abnormal, very strange crease, but it's very symmetrical. It's more symmetrical in the way it's put together than is the human skull. That is a typical human skull. All of our skulls are asymmetrical. Starchild is very symmetrical, surprisingly symmetrical. Next slide. Now, the rear view, the sutures and the inions, you see how the difference in the rear of the head is. This is the same pretty much volume of bone here for the occipital, but it's like somebody took it and just pulled it up and straightened it out. And this is the same three-point junction of the cranium. You know, do you know about the plates of your brain when you're born? These are separated by and held together by ligaments so that you can squeeze out your mother's birth canal. Your head turns into a tube while it's going out, and then it comes out and very quickly it expands up, and over the next 2 years they fill in with these things like this and they seal. And that's in 2 years, and the last part to go is the part up here, the soft spot, the fontanelle. Okay? So that's how that works. Now the inion, again, human inion, bump, a dent here. You can feel it. It's an absolute indentation where there should be an extrusion. Next slide. Okay, the balance points of the cranium is very interesting. With a human human skull rather, the face is imbalanced because we have those deep eye sockets, we have big frontal sinuses in here, we have hollow noses, hollow mouths. So it's like a fat kid and a skinny kid. You've got to move it. You've got to move the fulcrum back in favor of the skinny kid to give them more leverage. So our necks are centered. You can just see mine or anybody's back of center to make it work right. The Starchild is completely different. Its balance point is dead center down the middle because you got all this here and you're missing this back here, and you got all this here and you're missing the air up front because you've got that very shallow eye socket. You have no sinuses, which you'll see in a minute, and you've got a very small lower face. So the fulcrum has to be moved over and the center of gravity has to be moved over such that it's dead center. You take a cricket bat and you hold it down at the handle and you hold it absolutely straight vertically. Any of you can hold it like this very easily. You move it 2-3 degrees off of dead center and suddenly you start to strain because the weight suddenly increases. We have that with our heads and our necks. This is balanced on that skinny little neck like the cricket bat held absolutely vertically. Next slide. This is the brain, the different pressures on the cerebellums. Now, your cerebellum is an area back here about the size of my fist, in the back of your brain back here, and that is you. This is your software. All this stuff about your thinking capacity and all that, you need that, yeah, but this runs you. This is your urine, your bowels, your heartbeat, your blood pumping. This is your software that never stops running. This is you. You hurt that, you're dead. Russians, they execute you and kneel you down, one bullet in that, gone. That, the cerebellum, you just can't go without that. So with this one, as you see, with the human, all this 3-pound weight presses down on this. Well, this is designed in such a way that it's in the curve here because it has to hold up for the course of your life in good shape. It can't allow this pressure to come down and squeeze it out of the neck hole because you'll die if that happens. So it has to be set in place. So it's set in the curve of the back of your head, and there are ridges of bone in here that it sets and holds up against, so that it's built really, really well to do what it has to do. The Starchild doesn't have that. Those ridges are just down to nubs. They're not really there. And you have all this brain back here pressing down this way, so it's like a fat kid going down a slipper slide. It should just be swoosh right out of the neck hole. So when the first brain guy saw it, he was doing all this, he says, this thing either doesn't have a cerebellum like we have, or whatever its brain material is made of, it's 3 or 4 times harder than ours. It's like much more gelatinous than ours. And our brain is pretty doggone gelatinous. It's got to be really, really hard to stay in this head at this angle without coming out of the neck hole. You with me? So again, everything different. Next slide. All right, now this is the underside and the neck and what it all looks like. Now this is called the basilar part, and that doesn't really seal in individuals till we're about 25 years old, 20 to 25.
Wow.
Yeah, so this is kind of loose until then. So this is an adult, it's sealed, but the Starchild lost theirs. Now, this could have meant it was younger than the sealing age, or it just could have bumped a rock and just knocked it out in the tumble in the water. But nonetheless, the basilar part would be there. Otherwise, everything's the same. It's pretty much the same size.
What's it for?
The basilar part is just part of the brain holding. It's a very stout, thick bone that, again, is keeping pressure off of this in the event of a hit in the front. Because if anything happens to this, you're toast. So you've got some protective. You remember the things we were just talking about, the typhoid plates that they are. So it's just part of the bracing mechanism of protecting your.
You've got any ideas about why it doesn't complete itself till 20? It's interesting because I've always thought people aren't physically completed till they're 25. Keep telling my daughter.
I'm sorry, you're the first person to ever ask that. I do not know, but it's probably look-upable.
Okay.
I'm sorry I don't have an answer for you. But what you really see is the difference in the shape and the size of the neck. It's about half the volume, and it's an oval rather than a circle. Very different. Next slide. Okay, the areas of neck muscle attachment, this is what we were talking about a minute ago. You have the indentation here of the inion. The neck muscles should be attaching up here if they were human, but you see the fossa down here, and you have this little area of attachment. Big area of muscle attachment here. You can see the width of the fossa right here, which are down to this here. And so you see the size of this, and this is the inion. So just redesign, a complete redesign. that's so bizarrely different, and yet it's functioning, it's living. It dies as an adult. We don't know how old. Next slide. It's not a child. The zygomatic arches, cheekbones, and you see just this is a draw-in of what they normally look like, and this is the Starchild, and you see how much compressed the lower face is. All the signs of it are. Next slide. This is the chewing muscles that come up under the cheekbones, and they spread out like this, and all of us, you chew a little bit, and you can feel them moving right up there, and the Starchilds are right here. About half the volume of chewing muscle attachment. Again, a clear sign that the face is very, very reduced relative to a normal face. Next slide. Now this x-ray showed the Starchild lacked any vestige of frontal sinuses. You see the frontal sinuses in the human, that's the eye sockets right there. And you see in the Starchild, its eye sockets, and you don't see even a vestige of it. Normally when a person is born, even if they're born without sinuses, and you can be born with one of these bigger than the other one, you can be born with screwed up sinuses, but you're just not born with nothing there. That's just, you know, your body tries to do it, even if it does it wrong. It knows to do that. So what your sinuses are for, it's very interesting. You think it's for helping you with disease and colds and things like that, because that's what the medicine companies tell you when you do commercials and all your sinuses. Not so. What your sinuses are really for is to make your voice resonant, to make your voice speak like a human. Because if you don't have sinuses, you talk like a robot, like one of those things like that. You have no resonance whatsoever, right? So, and you hear people that have damaged or very bad sinuses, and that's how they sound. They don't sound, they sound weird. So, it's beginning a sign that maybe the Starchild doesn't speak. The little bitty neck, the lack of the sinuses, the very big brain, this is an indication that maybe it doesn't speak. It communicates by telepathy, which everybody says that's how the Greys communicate. Next slide. All right, the CAT scan shows the Starchild's inner ears are twice the size, and this is just representative. It's the same thing, but humans are about this size, and Starchilds are just double as big. Inner ears. Now, why would you have, you know, your inner ears are for your balance. Why would you need bigger, better balance? Well, if you've got a big head sitting on a skinny little neck, you might need a finer adjustment to keep it where it is than just these big muscles that we have that can do that job. Maybe it's a more balanced, a more fine balancing thing, and that's what they would be for. It's the best answer we have. Next slide. Okay, now this is really cool. The exterior surface of bone. In a human, you have these things called lacunae. Now, we've all heard the story that our cells change out every 7 years, right? You know, in every 7 years, everything but our brain changes out. Well, this is how it does in bone. Bone is constantly coming up through these things and being recycled out, and new bone is coming in, and it happens through the lacunae. All right? All bone has that. Starchild doesn't have it, as you see. Now, if you're going to be a deformity, what? Where would that go? And you're going to live. Where would that go? Very important thing. And you see, it's just like little closed out nodes, but they're not functional. Next slide. All right. This is the mass spectrometry of the human bone, and you see that there are spikes in carbon, oxygen, calcium, phosphorus, oxygen, carbon. But kind of get this in your mind. This is human bone, the way it is set up chemically by, you know, the chemical parts of the bone. Next slide, Starchild. You look at the difference. Again, the calcium, the phosphorus. Look at the oxygen and the carbon. What is this? What does this look like? You don't know. It's collagen. The collagen of your teeth. Like your teeth are the hardest bones in your body because it has an overload of collagen. You know, when you have your teeth drilled and you have that horrible icky smell, that smell, we all know that smell, that's burning collagen. It's not burning tooth, it's burning collagen. And so what this means is that the Starchild's bone is solid wall-to-wall tooth. It's much, much harder than human bone. Next slide. This is the piece of maxilla, the attached piece of maxilla, and that's this piece up here. And you see that it had a nose opening, and you see the two teeth when there were two teeth. You see it here like this. We eventually took this tooth for testing, a big mistake, big disaster. But anyway, nonetheless, this is how it looked. Next slide, you'll see it together, and you see those two fused together to make this. But guess what? This is flat. There's no arch at all. This is flat. So it indicates that it has a small tongue or no tongue at all. And that goes again to the idea that maybe it communicates by telepathy. Next slide. Now this is that piece of maxilla after the tooth is gone, but x-ray so that you can see that it has unerupted teeth in there. And when we first took the first x-ray of this, the guy said, well, if you've got unerupted teeth and you've got teeth down, it's got to be a child, and that's where the name Starchild came from. Later we found out that the sutures were of an adult and that these teeth, as you'll see in a minute, are heavily worn. So this could be a being that, like a shark, when its tooth wears down, it can replace, which would indicate that maybe it has a lifespan of 200 years, 300 years, 500 years, we don't know. But it can have a very extended lifespan, and if it did, and if it eats like we do, and it seems to, it's going to need this over time. And there it is. Next slide. So, and here you see this tooth when it's pulled out, and it's clearly not a kid tooth, but you also see this. This is crazing, what's called crazing. It's cracking of the enamel before death. If these are white, you know it happened post-mortem, and it's just the tooth shrinking up as you die. But if it's stained by food, berries, whatever, you know that this is crazing in life. And it's hard. You gotta like chew ice for 10 years to crack this. But you can also see the chips, where they're eating food that's stone ground with a pestle, and it's leaving little pieces of grit, and they're biting on that. But it's an adult. The whole point of this is not a child's tooth. It's an adult. Next slide. Okay, and this shows the cutting. Remember I told you that there was the cutting done and all that, and it's pretty extensive, and it's unfortunate, and there's stuff cutting on the top of the head too, so I hate it, but we've still got the majority of them, as you can see. We've got like 80% left or more, but we have had to. But look at you get a good shot, a good difference of the depth of the eye sockets. Very, very different. All right, next slide. How does mainstream science attempt to explain the Starchild? Well, they say Mother Nature can do anything, create any kind of deformity or mutation or variation, no matter how bizarre, unlikely, something looks. Nature is fully capable of it. I don't know why that's not there. But that's their answer. That when we would bring these physical differences, these incredible physical differences that would make any real, normal, natural scientist jump up and down and say, boy, this is really cool. We need to find out what this is. This is not something they want to know. This is not something they want to dig into because they know it's going to upset a lot of apples. And so they just say, well, nature can do anything. And I had them tell me that. When I would say, all these, they said, look, I don't care how many of these things you bring me. I don't care how many you bring me. All I have to say is nature can do anything and we win. And it's no different than religion where they say, I don't care what you say. All I got to do is say God can do anything and I win.
Exactly.
You know? Nature is the God of science, and they use it the same way religion uses God. They do when things they don't want to deal with. That's their go-to guy is God, and their go-to gal is nature, and that's unfortunate, but it makes it a very, very hard row to hoe to get anywhere with these people because they've got you beaten with that. Next slide. But we can work around them. We've got 10 experts here who officially state the Starchild is not a known deformity. And then there's more down on the bottom, one more on the bottom. But there you go. And these guys are on the Starchild having done a report. And the thing about these guys is they all saw the skull. They all held it and examined it and gave their opinion. All the people that you see on the internet that are like saying, oh, it's just a human deformity and all this, you've never seen it. They're just going from reading one report of somebody who said they didn't see it, and they say, I read a report that says it was not real, and so I say it's not real, and like that. Next slide. How do internet critics, skeptics, trolls explain away the Starchild? It was cradle-boarded, it suffered from hydrocephaly, it suffered from progeria. Those are the three things that they will tell you. Now, we're going to go over each of those and see if that's right or not. Next slide. Cradle boarding, typical cradle boarding, and an x-ray showing effects on a human skull. Cradle boarding is when native peoples strap a baby onto a board and the woman carries the baby on her back so she can go about doing her daily work. Well, when you're born, and the human that was with the Starchild was indeed cradle boarded. For that area was very typical in primitive societies 900 years ago. I should have said the Starchild we found was died. Both of them died. They've been carbon-14 tested to 900 years ago, so this was not uncommon. But what happens is when you put a baby on a cradle board, and you have to strap its head down. You can't just go doing your work like this. The baby's head is the biggest part of its body on a very weak neck, and it'll break its neck or hurt its neck for sure. So you have to strap the head down. And when you do that, you just put a little band around it. The bones are so soft, you're going to get a flattened area about the size of my palm on the back of the head, right down to the end, beyond which it won't go because you've got muscles starting right there. And you'd have to tear the muscles off to go any further, right? So this is what cradle boarding looks like. Now, next slide. This is cradle boarding, and this is the Starchild. Now notice the big difference. The convolutions are there right through, the natural convolutions of the bone. They're not flat. The human that's as flat as a board, a table, a floor. But that's not, and you can see the convolutions right here. Now, a moron can see this. A moron can see this and know that it's not cradle boarding. And yet, you go right on to Wikipedia right now, and you will see that it's a cradle-boarded hydrocephalic, or hydrocephalic for sure. All right, next slide. Next slide, yes, sorry. Now, this is another thing about cradle boarding, the positions when cradle boarded. This is how the heads are set up. Well, to be cradle boarded, this guy fits perfectly sitting there, and it's, just grooving, chucking, no problem. Put the Starchild in that, going to die in just a few minutes. Again, a moron can see it. Next slide. Now, hydrocephaly, different thing than cradle boarding. They always say it's a cradle boarded hydrocephalic. Hydrocephaly, we all know water in the brain. Well, the water comes in and it goes out in all directions. It's like blowing up a balloon. Blowing up, it doesn't differentiate. It blows up. It's fluid, and it's fluid coming in, and it's got to go somewhere. So it's, for an infant, these aren't sutured. These aren't, closed together. They aren't, and so they'll spread out, and they do, and it just gets bigger, bigger, bigger until the kid dies. What happens is the pressure pushes the brain out the neck hole, like we were talking about, and that's what it'll eventually. It doesn't just blow up like a overblown balloon. It just squeezes the neck out, squeezes the brain out through the neck hole, and it dies. Next slide. Here's another one, and you see, now you could say that the Starchild might have hydrocephaly if this was, because you can't blow a balloon up and have a crease in the balloon. But if this suture line was fused, then it could. If it was prematurely fused, and kids can be born, I don't know if you've ever seen those kids, they don't have them anymore, they had one in my community. When they're born with this thing fused right here, this suture fused, they grow up and their brain grows forward and their eyes go out to the side of their heads. You ever see one of those fish-eyed kids? They would institutionalize them and have to do all kind of surgery to make it go right. Really sad case, but they fix it now when they have the problem. They can suture it, make a shunt, and it just doesn't happen anymore. But if this were to have been fused, then it wouldn't have allowed it to grow like a balloon. But, next slide. What we see is that it wasn't fused either here or here, because this is the inside. We got a camera up inside shooting this way of this place right here. See it right there. And you can see the light coming through in different spots. It's clearly not fused. It's not just blocked like that. It's normal. This is normal suturing. That is too. Next slide. Now, last but not least, look at the vein tracts. When your brain presses up, you could say, okay, there's two kinds of hydrocephaly. There's water in the brain, and there's water on the brain. Water on the brain being the more common. But you can have sometimes water leaking out in the brain. But water on the brain is the normal standard thing. Well, you see here, you've got these vein tracts where the brain presses up into the soft bone and leaves the tracts. Perfectly normal. And you see the same thing with the Starchild. They're just in a different place in a different way, but there they are. No water on the brain. Moron level. Moron level. And mainstream science can't figure it out. Next slide. Okay, progeria. This is their new favorite because really, I make them pay for that cradle-boarded hydrocephaly. I make them look really stupid. So they've come up with this one as an alternative, progeria. Now, in typical progeria victims, their bones do thin out. Because progeria is that disease where you're born and you immediately start getting old. You start aging rapidly and dramatically, and you die about the age. If you make it to 15, you have really done well as a progeria victim. All right? So as you can see, this kid will be, 3? And it's already clear that he's on his way, unfortunately. But that's true, the bones do thin out. So this is why the mainstream jumped on it. Well, the Starchild has those thin bones, progeria victims have thin bones, but they don't take into account, next slide, that the Starchild's bone is so much harder. Now yes, it is thin. Look how much thinner it is. And yet, when you cut this, and I have with a handsaw, you can cut through this, and it isn't easy, I mean it's bone. But this is like cutting sheet metal by comparison to this. This is really, really hard stuff. Hard stuff, 2 or 3 times or more. We don't really know. We don't know how to scale it. I can just tell you from cutting it. And everybody that's cut it, I'm not the only person doing it, 5 people have cut it, all say the same thing. God damn, that's hard. And it is. I mean, there's just no other way to say it. It is really hard. Okay, next slide. Progeria victims have greatly reduced lower faces, and so does the Starchild. Now, this is another thing they say. Well, it's got the thin bones, it's got the thin face, progeria victim, right? Next slide. Oh, you'll see this in a minute. But it's not the same skull shape. It's not the same skull at all. It's got the normal brow ridge and nose. It's got the inion back there. It's just not. It's not. A progeria victim is still a human. A very unfortunate human, but a human nonetheless, with all of the human traits. Their eyes will go into the depth. Their eyes don't do this, or their eyes would bug out like frog eyes because there wouldn't be any room in there for a normal eyeball. So you got normal eyes, they got normal human physiology that the Starchild just doesn't have. Next slide. And last but not least, progeria victims nearly always have an open fontanelle. It doesn't close. And of course the Starchild has a perfectly normal fontanelle. Not progeria. Next slide. How do internet critics, skeptics, and trolls explain away the Starchild? It was created, avoided, hydrocephalus, progeria. And if that one rolled up a little bit, you would see none of the above. The answer is none of the above. None of that's true. None of that. Next slide. So if the Starchild skull was not a deformity or a combination of deformities, and it was not caused by typical cradle boarding or hydrocephaly or progeria, then what the, as my father used to say, blankety blank was it? What the hell was it? All right, next slide. This is an image by Roy Menzies, which is a fusion of X-ray and then drawing to fit what was there. A very clever way, I think, to go about doing it. Next slide. This is a cover image of the book Communion versus the Starchild by Roy, and again, this was his final product after he shaped that face. Now, this is the prototypical gray alien, but Bud Hopkins told me one time after seeing this, he says, this cover we use when we talk to abductees, he says, we'll put that in front of them and we'll say, is this what you saw? And if they say, oh yeah, that's exactly what I saw, he says, we know that person is probably stretching it because this was put on that cover to sell books. It wasn't put on the cover to be dramatic. In fact, Whitley Strieber himself argued to the publisher not to put that. He said, but that doesn't look anything like them. He said, yeah, but it'll scare the hell out of everybody in this book. And it did. It's one of the most arresting book covers. It just sold millions of copies off of that cover staring out at you out of a book rack. And so Bud says this looks much more like when we hear them described by people who are genuine abductees, it's much more like this than like that. Next slide. So forensic sculptor Bill Munns, this is another guy, who made a clay model of how the Starchild might have looked when alive. And this is it from both angles. Next slide. Let's see. This is where we find out if it works or not. Yes! Animation of Starchild sculpture. And you see Bill putting it together. Now, the neck's a little, in my opinion, too big. The eyes are ridiculous because he had to cut the... When I saw it, I said, Bill, what are you doing with those eyes? Those eyes look like normal. I said, you couldn't put an eyeball in there, a human eye, because you have eyeballs to make. He said, oh yeah, you're right, the eye looked like a big frog eye sticking off there. It didn't work at all. And I said, well, what did you do? And he said, I just cut it in half. And I said, well, the eye isn't at all accurate. He says, yeah, but Lloyd, you can't have real human eyes in those eye sockets. If it's got eyes, they have to look something like this, So, and the neck's a little wide. He couldn't live with that little pipe stem neck. So it looks like it's just going to break off, and he didn't want to have the neck break off, bump it accidentally and break off or something like that. But anyway, so now hit it again. Animation and redux, now that you know about the eye. Now watch the half eye socket. You see the half eyeballs in there? And the little neck he fleshes out. But that's still pretty cool. Now, a lot of people ask, you know, me, do you think it's possible that the Starchild could ever, you know, be among us, or somebody like the Starchild? Would we know it? Well, next slide. And I guess, oh, excuse me, I'm sorry, I got ahead of myself there. Proportional analysis, no, that's it. You were okay. Right here. This is really cool, and I forgot that I added this just recently. Proportional analysis of the Starchild face by Chris Murphy. And what he says is that our faces are really built interestingly. Our eyes are at the 50% mark. The tips of our nose are at 25% of what's left. And then our chin, our mouth is at 12.5%, 12.5% to our chin. Our faces have pretty good balance within percentages. The Starchild's nothing like that. It's 57% down, 11% instead of 25, 15% instead of 12.5, 70% instead of 12. Anyway, it's very, you know, different. The facial proportions are very different. Now the thing was, can they move around us? Yeah, there you go. Next slide. Can they move around us? And, I had Vivian to thank for that. She came up with that.
Yeah, but Paul McCartney, it's the wrong Paul McCartney.
Yeah, that's right.
It could be the wrong one, yeah.
Not the real one in the middle.
Next slide. Okay, two things found in the Starchild bone. Now this is really interesting. Two things found in the Starchild bone are not found in the bones of any other creatures on Earth that are known at this point. Unknown fibers are embedded in the matrix of its bone, and unknown red residue is in its cancellous holes. You'll see both. Next slide. This is the fibers, and this is the cancellous holes, and this is just a piece of the bone that you saw that we cut out, that you saw. And when you look at it under a microscope, this is what you see. You see these fibers dangling out of the holes and strung around it, and they are unbelievably durable themselves, because that's a Dremel blade. You see these things here? That's a Dremel blade doing it, you know, making these cuts. And yet they're resisting the cut of the Dremel blade. So that tells you how strong they are. And they are embedded in the matrix of the bone like rebar through concrete. And we're going to see some up close. Next slide. We'll see this knot up close. Next slide. You see this knot here, that knot there, and then we see these two over here up close, and we see that they're different. But here you see, what's important to see is it's coming out of this track of the bone here, and it got snapped up here, and it seems to have been pushed up through here, and this thicker kind got wrapped around it in a way that we can't imagine. It's like how a magician did it or something, I don't know. But anyway, and you see a different kind, but these things are in the matrix of this bone. And what they do is they add to the strength of the collagen, the heavy collagen, that's reinforced with that. Why does it need such strength in such thin bone? Who knows? And it might have to do with intergalactic travel or something like that. We do know that when our people are up on the space station, their bones weaken, and one of the first things that happens when they come back is they find that their eye sockets have shrunk a bit. Mm-hmm. Everybody comes back seeing a little different if they've been in there 3 months or more. Their eye sockets start to shallow to humans. And you see the Starchild with its shallow eye socket. So there's something about being in space that does that. Really weird. Okay, next slide. This is another isolated flat fiber. This is yet another kind of fiber. We just don't understand these fibers, but we do know that they didn't just grow on it because the bone was, it was just come off the thing and they were there. So we had mycologists look at it. It is not anything bacteria, fungi, mold, nothing like that. They're there. And this is, you can see a frayed piece right here where this piece got hit by the Dremel blade. Instead of cutting it, it just frayed. Strong stuff, whatever that is. Next slide. And this you see a perfect shot on the inner part of the bone of it lying in the matrix, so that there's just no argument that this stuff is in the matrix of the bone. Next slide. Now this is the reddish residue we're going to look at. And this is chips of bone cut but not polished. This is where you cut them, but we haven't polished either one, the human or the Starchild. And you see how much milkier it looks because it's got that much higher collagen content. Next slide. You see the human bone when it's polished, and this is the same piece of bone, but that hole is that hole, this hole is this hole. So you see, and that's the outer, and this is the inner surface of it. But you see that in the course of death, when you die and when you're decomposing, in the natural course of events, you are carrying bacteria in your body that is designed to wait until you die and clean you out. Just clean you out. And it will clean out your not your collagen, your, what's in there?
Marrow.
Marrow, sorry, marrow. Thank you, Jerry. Thank you. It will clean out your bone marrow like you see right here, to the point where you can eat out of it. I mean, it's just beautiful. This was, to me it looks like alabaster, but it was very carefully polished, like with lapidary tools, like jewelers polish it. And this is what you get. And it's really nice. And this is how you see, there's not a shred of marrow left anywhere. It shouldn't be, and it isn't with the human. Next slide. And with the Starchild, it's completely different. You've got this reddish residue all over the place. Now, that's not going to be blood marrow, because that's blood, and it's going to be black if it's if it's oxidized. It's something else, and we don't know what it is, and we just don't have the money. The problem with what we have is we have this incredible thing with all these neat things to find out, and we can't get the money to do anything with it because science doesn't want this answer. I've had this stuff out for 13 of them, my 14th year now. Any of them could have come forward and said, we want to know too. They don't, because they don't want to know. They want to have me floundering out here as long as they can keep me away, they will. I'm going to beat them eventually. I know I am, but it's been a hard road. But this stuff is what keeps me going because I know it's for real. Next slide. Here you'll see another shot of it, backlit, so that you see plenty of it. It isn't just a fluke thing. Something going on here. We wish we knew what it was, but we don't. Next slide. Everything about the Starchild Skull's case comes down to its DNA. DNA is a molecule of life. This is a cell, and inside the nucleus are the chromosomes. Chromosomes come down and they unravel. You unravel the chromosomes, and you're looking at the DNA. And the DNA is made up of the bases. We're going to see that in a minute. And what the DNA does is the good, not the junk, the stuff that works, makes proteins. What you have to understand is about 97% of all of this, of everything in here, 97% is junk or non-coding or whatever you want to call it. It doesn't make the proteins. About 3% of this runs us. The rest of it is like, what is it there for? We're still trying to figure that out. It could be spirit, could be a number of things. But junk is not dealt with at the scientific level because it's not quantifiable. Okay, they can't quantify it. So anyway, this, we need to all understand this, and we're just getting started. Okay, next slide. We'll see a different look at it. What we were looking at is made up, the base pairs are only 4. There are only 4 base pairs. Thymine, adenine, guanine, and cytosine. And that's it. And these two go together, thymine, adenine, and guanine, cytosine go together, and they don't mix. It's these two, and they make these steps, the base pairs. These are the base pairs. These are the nucleotides. These are the base pairs that the two nucleotides come together and make. So that term is used kind of interchangeably. When you say a nucleotide, if you know the nucleotide, you know what its partner is automatically. So all you need is the nucleotide, and you know it's a base pair because you know what the other side of it is going to be. So you can strip them apart like this and get to nucleotides, and you know what goes on the other side of it. So nucleotide base pairs, don't be confused by that. It's essentially the same thing. We tend to use base pairs, but if you see nucleotides, not a problem. So you have all of this, and all 3 billion of them are wound up like a wad of rubber bands in the nucleus of the cells that we saw just a minute ago. They're just like rubber bands, just wadded up. And that's it. Very, very tightly packed. Next slide. All right, each human cell except red blood cells has nuclear DNA and mitochondrial DNA. You need to understand this. Mitochondrial DNA, this is the cells in our body, all of our cells except our red blood cells, have a nucleus. Nuclear DNA is found in the cell nucleus and contains genetic material from both parents. Mom and dad make the whole genome. Mom supplies her 23 chromosomes, dad supplies his 23 chromosomes, and you get 46 total. That is, each cell nucleus has 3+ billion, keep this in mind, 3+ billion nuclear DNA base pairs. 3+ billion. Now, mitochondrial DNA, that only comes down from your mother. Now, the mitochondria float outside the nucleus, but in the cell cytoplasm, like raisins in pudding or something like that. But there can be hundreds to thousands of them, depending on which cell we're talking about here. Now, that comes, mitochondrial DNA is found in the cell mitochondria. Now, these are the energy points. These are the power plants. These make the proteins and do, this does the work. This just sits and is sort of king of the hill, but this does the work. It contains genetic material only from the mother. Why? Because an egg has plenty of room. You've all seen films of the little bitty sperm coming and hitting the egg. So the egg carries the mitochondria. The sperm is nothing but just the DNA package and a tail. The egg has her DNA package and all the mitochondria. So it only comes down from the mother. So the mother, the grandmother, the great-grandmother, the great-great-grandmother, right on back, they all have the same mitochondrial DNA. I have the same mitochondrial DNA. You have the same mitochondrial DNA of your mother, your grandmother, your great-grandmother, going on back with very little change. Okay? And so that's why all human mitochondria contain 16,569 base pairs. Sometimes one up, sometimes one down, sometimes 70 or 68, but the number we nearly always share is 16,569, precisely, all the time, and each cell holds thousands. Now the difference is, this is swapped around from the nuclear DNA. The nuclear DNA is 97% junk, 3% working, right? This is 97% working, 3% junk.
Wow.
So this doesn't change much. It doesn't allow much room. You can have a lot of mutations in here because the odds are it's going to hit in the junk and isn't going to have a real major impact on the functioning of the body. But here, you get a mutation or a change or a difference here, and you hit out in that 97% that's working, you're going to have problems. And if you have faulty mitochondrial DNA, don't have kids because they aren't going to make it. That's where if they find women who have faulty DNA, they have 1 or 2 or 3 miscarriages and they figure it out, they start taking their genetic package out, but they'll take the egg of a woman who doesn't have the mitochondrial DNA, so their kids will be born with the genetic package of mom and the genetic package of dad, but the mitochondrial DNA of a third woman. But you can have your own children that way. A couple can have their own children. Very cool how this works. But anyway, all right, next slide. This is early DNA testing used primers, 20 to 30 base pairs long, below is an early array in a gel sheet. Now this is important to understand because you have to understand the story of the Starchild's early DNA testing. Next slide. The first testing was done at the University of British Columbia. I made a mistake. We couldn't, there were only 6 labs in the world that could do nuclear DNA at that time. None of them would touch the Starchild. And so there was this lab here. It was a new lab. It was a dental forensic lab, totally incapable of dealing with ancient DNA. And the guy said, well, I'll just try to find out if you have live DNA there. Because sometimes a skeleton bones can be so degraded that they just can't recover anything, but either mitochondrial or nuclear DNA. He says, I can't recover mitochondrial, but I can recover nuclear, and if I find anything nuclear, you'll at least know it's worth trying to gather the money and keep going. So I said, okay, great, next slide. So what he came up with was after 2 times where they contaminated it, and a 3rd time, they came up with this result. Now by then, a guy on the team, one of the kids there, again, this is a university, this is a main geneticist and kids and students doing the work. And they're just screwing it up. They're just contaminating it every time. The first 2 times, and they did it again this time too. But by then, the rest of the university had heard what they were doing, that they were working on maybe an alien skull, and the other professors dropped down on them, and now they were just trying to get rid of us. Get us off the camp, get us out of the deal. So he said, after the customary number of PCR cycles, 28, there was a very weak 200 picograms gender profile from the second bone sample taken from the child's mastoid process. Now, normally 1,000 picograms were the usual minimum threshold at which you could tell anything about what you had. And the guy says, we only got 200 picograms, but we know that it's a result as an XY chromosome, which tells us 2 things: the child was male and it was human, right? This is a totally BS report and a totally BS answer. But the problem was in late 2099 that went out as the official answer, and it was exactly the answer that mainstream science wanted. Human male. End of story, end of tape. We don't have to worry about the Starchild anymore. And so to this day, you will read that this is the answer, that it was already proved to be human, even though this was in 99 when they were using primers of 20 to 30 base pairs long, and it was easy to make mistakes, and these guys didn't even get it. They didn't even get it because why do we know? The next slide. Now this, you have to understand, this caused a disaster to our team. We couldn't get a hearing anywhere. The Bowl lab report equaled 3 years of dead time. 3 years from November of 99 to November of 2002. When, by that time, I had spent so much time and energy and effort trying to resuscitate it, I had gone horribly into debt, about $50,000, and I was going to have to go bankrupt. So my girlfriend at the time says, "This is killing you. This is killing you. You have got to stop this. You've got to just give it up." So I sent out the word that I was, I just couldn't go anymore. I had done my best, but somebody else was going to have to take over and go from here. And then, next slide, Miss Belinda McKenzie of raises the dead. Raises the dead. She contacted me, first contact, "What do you need?" "I don't know what I need. I haven't even given it any thought for so long. Find out what you need and let me know." And so we did, and we found out, and we told her, and she did her due diligence. It took a few weeks, but finally a check arrived and we were back in business. So Belinda is one of the key and major players in this whole thing. Without her, I wouldn't be here today, and that's the truth. So again, all right, next slide. Yeah, so what she paid for was a real DNA test from real geneticists who were capable of recovering ancient DNA. Now, what do I mean by ancient DNA? Older than 50 years. That lab in Vancouver, they only dealt with fresh DNA. They couldn't deal with very old DNA, much less 900 years old. Over 50 years, there's a huge amount of degradation, and there's a huge amount of contamination from the bacteria of death that we were talking about. Right? So this is not an easy thing to do, but these guys could do it because they had worked on the Kennewick Man. Anybody heard of the Kennewick Man? The Kennewick Man. Yeah. This was a skeleton that was found in Washington State on Indian territory, and it looked clearly like a normal European. And the Indians went bazookas because they have these gambling concessions in the States, because they get all these special privileges because they're supposedly the first people to settle America, and so they work really, really hard anytime something comes up to indicate that somebody was there before they were there. And the Kennewick Man was like 9,000 years old and looked like a European, so that wasn't going to happen. So they made the Clinton administration cement the whole thing over, and the Kennewick Man, and it became this big legal case, and they eventually, it took like 6 years to be able to study it. Well, these guys studied it, and what they found was that the Kennewick Man was exposed on the riverbank. That's how it was found, and the water would wash in and cover it, and then the water would go out, and the sun would bake it, and so it was absolutely sterile. There wasn't any nuclear DNA, there wasn't any mitochondrial DNA, there was just nothing to recover from. So at that time, later they were able, as the refinements got better, they were able to tell its date and stuff like that. But at the time, that's where they were. But these guys did a great job, and here's what they did. Next slide. Using, again, primers. Keep in mind, 20 to 30 base pairs long. 20 to 30 base pairs long, short-length base pairs. They recovered the human's mitochondrial DNA, and she was from haplogroup A, and they found mitochondrial DNA for the Starchild from haplogroup C. And that's what that means. They also found, next slide, that in 6 attempts at finding the nuclear DNA, they couldn't do it. They were 10 times, 100 times better than the lab in Canada, and they couldn't do it in 6 attempts. They could not find nuclear DNA. So that proved right here that what they had said in 99 from Vancouver was wrong, just was wrong. Should have been thrown out right there. Again, you can still read on the internet that the 99 test was better than this test. And this test has turned out to be wrong because those primers are so short, 20, 30 base pairs long, and in 3 billion base pairs, it's not too hard to find pieces 20, 30 long that match up. But when you get out to 2 and 3 and 400 and 500 long, it's a different story, as we will see with the new geneticists. Next slide. So, but out of trace genetics, those two guys, the result was that the Starchild Skull was a human-alien hybrid, and some of you might remember who have been here when I talked about it as a human-alien hybrid. You might have seen that very same slide. We believe that up from their test in 203 right up until 210, we believed that was the result. Next slide. It was what the guys told me in 203. I said, well, what can we do now? And they said, well, you're going to have to wait, if you can believe this, 3 to 5 years for this new technology to come on board that instead of primers, it's going to do it base pair by base pair by base pair. And that's what you need, because if your thing has never been seen before, the base pairs are how it has to come out. You have to be able to pull it out base pair by base pair. Sure enough, almost three years to the month. That was July 203. This is July 206. A company called 454 Life Science announces that they have a technique for doing it. I'm not going to go into it. It doesn't matter. It's impossible. It's incredible. It's unbelievable how they do it. But they do it. They can pull it out. They can disassemble DNA base pair by base pair. It's just simply astounding. And then tell you what you have. Next slide. So, in early 210, a new geneticist contacted me and offered to help using that new kind of equipment. From 206, the first few of these things that were done, the first few genomes that were recovered were like $10, $15 million each. The price just was plummeting that whole time. So now it's down to maybe, to do ancient DNA, it's just a couple million. Now, a lot of people say, well, wait a minute, why does it cost so much to do the Starchild DNA when I read on the internet that I could, you can have your DNA done for $5,000, $10,000. Well, that's true. You can have your DNA, your genome sequence for about 10 grand right now using this equipment. Because it's fresh DNA. You saw the rubber band pile, all these things do is they unbuckle the rubber bands and just string them out and measure them. Fresh DNA, easy to do. Now, imagine when you're dead for a long time, imagine that wad of rubber bands put through a wood chipper. And now all the little pieces. And that's what you've got to put together to make the genome of something that's been dead for a long time. And that's our problem. To do that, you have to do it again and again and again and again, 50 to 100 times. Run it through the machines 50 to 100 times so that you fill in every gap and you string the rubber bands back together. Basically, yeah. So that's why it costs so much. Next slide. Anyway, when he did, after a few weeks, he recovered and sequenced dozens of fragments of the nuclear DNA. That's all he got the first time. Dozens of fragments, but they were long, but they all came out of the junk. But they were in the hundreds. And so what he did was when he got those dozens, he sent them all to what's called the NIH database. Next slide. The NIH is the National Institutes of Health in Maryland. And what they have is, in America, they put out the money to most labs to do DNA work. So that if you want to do some kind of work, they will give you the grant money to do it. And in doing so, you owe them the results. And they take the results and they put them in the database. So by 210, when we were doing this, middle 210, the database was very, very full of literally billions of coherent base pair strings to compare to. All of the human, all the chimp, all the gorilla, and parts of hundreds of other animals. So it had a very, very good spread, down to bacteria. And yeast and molds and things like that. Everything anybody did, it went into that database and it was there. So if you had unusual DNA, that's what you did. You sent it there to see if it matched up with anything. Next slide. So what the Starchild did was some of it did come back in strings that matched up in the human genome. Some of it did. This is one of 265 base pairs. Okay, and it says very clearly that 265 base pairs is found exactly matched in Homo sapiens chromosome 1 genomic contig here. They know where it is, right? But half of the things he sent in, next slide, half of them came back. This is a 342 base pair. No significant similarity found. Not on Earth. So he contacted me and he says, boy, you really got something here. He says, we can have a few of these maybe, but not half of them. He says, you've got something here. Let's go to work on it. And I told him, we don't have a pot to piss in. I'm sorry. But we can't get the kind of money you need. And he says, let me think about it. And that's when that plan of keeping the little pieces came up. To do it, to do it more seriously. So he began to do, and he did another one. Next slide. Oh, excuse me, this is the next test he did. He was so excited, he did this test. And this is really cool, I think. Spectrophotometer illustrates light wave absorbance in pure DNA. This is how it looks, human DNA. This curve right here. This is how no DNA looks. And this is how protein contamination looks. And they all know how this goes. And so he just wanted to see how it would look. Next slide. And what you see is the human looks very much like it's supposed to look, but the Starchild looks like kind of a drunk snake, doesn't it? Just, what is it? It's not really no DNA, it's not protein contamination, it's not human, what is it? And this too, he said, this tells you, this is very unusual what you have here. I don't know what it is, but this is not normal. Very cool. Next slide. So, now let's go back and get back on track because we're going to start talking about mitochondrial DNA now. All right? So you've got to get back where we were. Remember, 3 billion base pairs in the nuclear DNA and 16,569 base pairs in the mitochondria. But that's where 95% of it works. That's where you don't get much change over time. And only females pass it on, etc. Next slide. So he recovered, in one of his next attempts, he recovered 4 fragments, 4 from mitochondrial DNA. 4. And the problem is, if it comes in junk, you can't figure anything out. You can't compare. You can't do anything. You've got to get it from the working parts to be able to compare it head up to what the human parts do. So, mitochondrial DNA is circular in design, and it is, and maintains astonishing efficiency, only about 5% is junk, which is more like 3%, that does nothing critically important to its functioning. 15 primary genes are very well understood. This is one of the best understood parts of your body. Each one of those, the RNAs, the NDAs, all that's very well understood. Encode for RNA. Mitochondrial DNA is extremely highly conserved. It undergoes so little change over time, those changes are used as the biological clock. You've heard about mitochondrial Eve. They take human females and they clock back their mitochondrial DNA, and the oldest DNA is found in black Africans of South Africa, just where Sitchin said the Anunnaki said they created us. That's where you find them genetically. And they're about 200,000 years old. That clock takes us back about 200,000 years to those black African women. Us, sitting here, 60, 70, 80, differences. Those have 120. The oldest of us have 120. We'll get to that. Next slide. All right. Human haplogroups. They're like 30, now you can sometimes get 33, 37, 38. People argue about the haplogroups, but these are the haplogroups right here. HPTA. Remember the haplogroup A for the human, haplogroup C for the Starchild, 2 basic ones, and we don't even know that that's their haplogroups, and these are the rest of the haplogroups. And here you see the human mitochondrial DNA from the Cambridge reference sequence. To get like the standard baseline, they picked one person and they did their mitochondrial DNA, and that became the baseline against which everything else was measured. Cambridge reference sequence, it was done at Cambridge. And then you have the Neanderthal mitochondrial DNA, and you have 2 of Denisova, the Denisova bone and the Denisova molar. Well, what is Denisova? Next slide. Surprise! Denisovans are a new addition to the early Homo pre-human family tree. If you haven't heard about them, they just got this finger bone and the tooth out of a cave, and they thought it was with Neanderthals, and they thought it was Neanderthal, but they measured its mitochondrial DNA, and its mitochondrial DNA was so different that they knew it had to be, even though they only had a little piece of finger bone and a tooth, it had to be another species. Just because mitochondrial DNA is so well conserved, and everything fits within such a tight window of each species, that if you're outside that window, you're something else. This is very significant. So Denisovans were discovered in 210. They seem to have been contemporaries of Neanderthals. Their DNA exists up to the 5% level in modern Micronesians, Fijians, New Guineans, and Australian Aborigines, and new groups being discovered. This is very exciting in the world of anthropology. Next slide. And biology and genetics. Now, base pair nucleotides, get with this, 1,255 of 16,000. Let's say we had all 16,000 stretched out here one by one. We're going to take out this narrow slice from 1,255 to 1,350. These 95 contain no variations in any haplogroup. They're clean. There are no changes. When we talk about highly conserved, that's what we mean. No changes over time in none of them. Very highly conserved across all 33 haplogroups, the one Neanderthal and the two Denisovans. Next slide. Now we're going to take another stretch. Oh, I want to show you what variation looks like. Variation in the human mitochondrial DNA. These lines come up, the computers do this automatically, but you see where everything else has a T, there's a C right here in this haplogroup. That's a variation. That is a variation. Keep that in mind. You can have them. You do have them. And over the whole course of the 16,000, humans have 120. Like that. Next slide. Okay, this is the other way they can be. They can be a change or they can be a missing. If there's a missing nucleotide, the computer automatically puts something in to show there's something different here. So you can either have a complete change or you can have just a missing spot. Up or down. You can see up or down. Up, down. Next slide. The computers know. Now here we have a different stretch. We have from 1426, from 1262 to 1426. This is, instead of 95 like the other one, this is 164 base pairs long. Now in this one we find that each of the Denisovans have one difference. You see it right there? The Neanderthal has one right here. That's C right there. And the human has one right here. That's C right here. Each of them have one. This is a very highly conserved area, but there is one change for everybody. Now we're going to see the Starchild's exact same segment right here. Next slide. 17 changes. 17 differences. You with me? Now right here, folks, this is a home run. Just right here. To show that it's not human, it's not Neanderthal, it's not Denisovan, it is something else. And Vivi came up with this great analogy that I'm going to use and going to share with you. It's as if the human, the Denisovans, and the Neanderthals, or at least in the States, this is how I know it. Let's say humans have an area code on their phone in Louisiana. Neanderthals have an area code in Tennessee. Denisovans have an area code in Pennsylvania. The Starchild's area code is on the moon! On the moon! It's that far away. You with me? It's that big a difference. Now we're going to see it in more detail, but this just shows you right here. It's very, very dramatic. Next slide. In the mitochondrial DNA of all humans, a maximum of 120 variations, again, a maximum. We have less, all of us in this room, are found in the oldest human type, Southern Africans. The rest of us have fewer. Neanderthals differ from the human CRS by 200 plus or minus variations. Denisovans by 385. Remember I said they have to call this a new species because it's so far away from 200. Yeah. It's 185 away when we're only 80 away from Neanderthal, and it's different. You see what I mean? Mm-hmm. They are more different from these than we are different from them. Mm-hmm. Are you with me? Now, chimps are out to 1,500. 1,500. What do you think the Starchild is? Next slide. You can't even imagine. 4 fragments of Starchild mitochondrial DNA add up to 1,583 base pairs. 1,583 is 9.5% of the 16,569 base pairs in the human mitochondrial DNA. In the Starchild's 1,583 base pairs, there are 93 variations, almost to the 120 maximum in just the 4 fragments. 9.5% times 93 equals 883.5 if you blow it out to 100%, if you extrapolate it out in the Starchild's mitochondrial DNA. the Starchild mitochondrial DNA will be 700% different, 700% different from human. This is, the slide is screwed up, as you can see. But here it is, between 800 and 1,000. Between 800 and 1,000 as opposed to that. Area code on the moon. Very, very different. And established, 4 fragments, 9.5%. With the mitochondrial, with the nuclear DNA, when it's 3 billion, all those fragments that he figured up, it's like 50 million, it's still peanuts. It's just a small portion. That's not a small portion. This is significant, very significant. Next slide. Now we're going to move to the big kahuna, all right? He was looking for something out of that 3% that worked, not mitochondrial DNA, but nuclear DNA. He started calling it the golden needle. I've got to find the golden needle in the haystack. One of these, it's called shotgunning what he does. He just shotguns it out and it can hit anywhere. It's like a shotgun blast. You just never know what you're going to get. It's a shotgun. So he says, I've got to find the golden needle in the haystack. I've got to find a piece that comes out of the 3% that works, so it will tell me something about how the Starchild works versus how the human works. So he calls me up and he says, man, guess what? I didn't find a golden needle, I found a diamond needle. You won't believe this. And this is what it is. In every human embryo, a gene called forkhead boxp2, or FOXP2, is a master gene controlling embryonic growth in at least 300 other crucial genes, and some people argue it could be as many as 1,000. So this one single gene, FOXP2, controls the effect, has a downward flow on at least 300 and maybe we don't know how many. They're still trying to figure it out. But it controls motor skills, we know for sure, brain development, lung development, speech, nerve communication. It's like your center core, and it's controlling all of that. So, what do you suppose happens if there's some kind of a difference or a variation or mutation in a FOXP2 gene when you're an embryo? You are screwed, glued, and tattooed. You are done for. You are going to miscarry, as a rule. One change, one. It's one of the most highly conserved genes in our bodies. Why? Because it can't afford mistakes. It just can't afford mistakes. Now, there are mistakes. There is one change that can be made, and you will be born, and you will live, and you will never speak. That's how they found out that it's speech related. So you can have one, but if you do, and you're born, as I said, you are screwed, glued, and tattooed. You are not in good shape. You need absolutely everything to go right in your FOXP2. All of us sitting in this room, our FOXP2s are the same, exactly. Every base pair in yours, and yours, and yours is exactly the same as every base pair in mine, or we're not standing here being who we are. FOXP2, highly, highly conserved. No change, understand. Next slide. Now, and the FOXP2 gene has 2,594 base pairs. It is one of the most highly conserved genes in our bodies. Changes almost never happen. Each one of those 25 in normal humans is exactly the same. What I just said. Next slide. This is FOXP2, what it looks like. A 211 base pair fragment. He recovered a 211 base pair fragment of the Starchild's FOXP2, the diamond needle. This is it. FOXP2 is compared to the exact same 211 base pairs in human FOXP2 in blue, this one, and in other species of animals. Rhesus, monkey, mouse, dog, elephant, possum, chicken, frog, and zebrafish. Right on down. Okay, and you see, exon, this is it. This is an absolutely head-to-head comparison. Remember I said he had to be able to make a head-to-head comparison? Here you have it, right here. What does this tell us? Next slide. What it tells us, this is too bad. Starchild's 211 base pair fragment is 56 variations. There's your rhesus monkey. There's no variations in humans and Neanderthals and Denisovans. It's all the same. In a rhesus monkey, you're up to 2, 20 for the mouse, 27 for the dog, 21 for the elephant, 21 for the possum, 26 for the frog, 56 for the Starchild. Really, really amazing. So 211 is 8%. It's almost like the 9.5% of the other. It is significant. of the base pair total in the FOXP2 gene, extrapolate 8% out to 100, you get 12.5. 12.5 times 56 is 700 plus or minus Starchild DNA variations. 700, where 1 is the maximum. You with me? This is what the, next slide, this is what the geneticist, oh, I'm sorry, I forgot. This is the newest one, I'm sorry. The newest one is he found a collagen-like protein fragment identified by a collagen pattern search. A human fragment sequence is shown in blue. The collagen characteristics pattern of glycine is pink. Note the amount of proline in association with the glycine. It has a special meaning. You see glycine, proline, proline, glycine, proline, glycine, proline. You see it like that all together? Guess what pulls up for this? Next slide. The glycine-proline pattern in the previous fragment resembles the pattern found in collagen 8, a protein found largely in the corneal endothelium of human eyes. Now, let me explain how this works. Collagen makes up 25 to 35% of our bodies. It's a lot of our bones and all that. Collagen, it makes up a lot of things. So there's a lot of different kinds of collagen. Collagen 8 is this very exotic kind that makes this lining of the inside of the eye that faces into the, to the lens from the cornea. Very, very specific collagen, and that's what he finds from the Starchild. Now, it's very, very different from the human because the Starchild has a very, very different eye. Another piece of the puzzle that's just completely different. So next slide. This is where, it's too bad we're going to miss some of that. The comment from our geneticist in his recently composed abstract. Now, he wrote an abstract. It's on the Starchild website. You can try to read it, but I assure you, you won't get very far in it because it's written in that language, that science language. But this is where the geneticist from Stanford contacted me. He said, I read the abstract and I'm very impressed, and I want to meet with you about it. So this is what he says. The probability that such a highly specific arrangement of changes in a small fragment could have occurred by accumulation of sequencing errors is extremely low, if not close to zero. Nor could this arrangement have occurred due to contamination with the DNA of any known species. From this evidence, one may conclude the underlying biochemistry of the entity's life form must be either the same as or highly similar to humans or other species, yet the use of the genetic code, which still, and basically it says, it's sort of the same, but is clearly very different. And again, this is more on level of what I've been showing you here. Anybody can see this. Next slide. So, where are we going with this? In the 1500s, these men moved the Earth from the center of the universe to its proper position. This was a profound experience for everybody alive at the time. The Earth is not it. The Earth operates, circles around the sun. Everybody had to adjust to that. It was shocking. Next slide. The Wright brothers in 203, they moved us all into our future where our planet gained a convenient scale. They shrunk the planet for us. They gave us a different world. Next slide. The Starchild is going to do, Neil Armstrong took the first step for all of us toward our inevitable future in space. Boy, that better be an important call because this is right at the end. Okay, anyway, so this too was a big, big step in human history. Next slide. And the Starchild Skull is poised to transform humans into members of a galactic community, to prove that we are members of a galactic community. And coincidentally, next slide, maybe the Starchild heralds a new beginning that we have all heard 2012 might bring. This could be it. This could be it. The new future, the new whatever. Instead of all what they're saying, maybe this is it. We still have time to be it if we get our funding, which could happen. Next slide. So, how do we get from here to there? How do we move the Starchild from being a partial proof of one of the greatest discoveries in all of human history to acceptance by mainstream science for what it so obviously is? How do we do that? We follow, next slide, we follow my friend Bob Rue's philosophy of life. Now, you all remember Hurricane Katrina. Here's Bob with, don't try. This is to the looters. Don't try. I'm sleeping inside with a big dog, an ugly woman, 2 shotguns, and a claw hammer. This is his Oriental rug shop. He's one of the guys that didn't leave. He stayed to protect what was there from the looters. And the water, I might add, you see where he's standing? About 2 feet from there is where the water rose to its high point. But he knew that was going to happen. Because what people don't know, and it's always a nice little anecdote to throw in, is that everybody wonders, why in the world would New Orleans build a city in a bowl? Why would that happen? Well, what really happened was back way in the 1700s when they were first settling the area, the Mississippi River would flood out to 100 miles on each side, and that's how the Delta was created, right? So the French and the Spaniards and people like that who would do this kind of stuff, they knew that they had to wait to the highest point of the flood, sail down the river, and find any land that was up and was suitable to build a city, because that would never flood. And there was this big crescent of land in one of the bows of the Mississippi River, and it was the biggest one there was, and on that crescent they formed the city of New Orleans. And it sure enough, it never did flood. That part is still the garden district of today, if you've ever been there, the rich part of town. But as the city grew bigger and spread out, it spread out into the floodplain, and that's what happened. And then they began to build the skyscrapers there, and it sank it down, and now it's in a 17-foot bowl as the soft land went down. You can't tell it just standing there. You just feel everything flat because it's like this. But Bob knew his shop was on the old, everybody knows where the old line is. And he said, it's going to come close, but I don't think it's going to, because Oriental rugs, Oriental rugs are ruined if they get, floodwater. And they're ruined if they get stolen, and he wasn't going to let that happen. So he stayed and he stuck it out. Don't try. This sign, by the way, became famous. It was in over 800 newspapers around the world. He became like a symbol of defiance of the looters and people sticking it out and all this. And that sign is now in a museum in New Orleans, and if you go to it, you'll see it. He was offered $10,000 for it by one of the main newspaper people that were there. I'm trying to think of his name, but anyway, what Bob did, I'm sorry, I'm just rambling, but it's a fun story. He and his wife, he looks like a complete slob, he is. He's a bum. He and I were roommates in the French Quarter when we were young. Did some damage to the girls, I won't deny it. But nonetheless, this is a cool guy. He doesn't look it anymore, I don't look it anymore, but we were neat in our day. He looks like a slob, but he lives in one of the Garden District mansions. His wife is fairly wealthy, and he does okay here, but this is just how it's like he says, he's a fisherman, he's a Greek fisherman's son. He says, I'm going to be a Greek fisherman's son till the day I die. So, he stayed, and everybody that saw this knew he was there, and they started calling him. And they were saying, Bob, go to my house, the key is under this, and go in and make sure everything is okay, protect us from the looters, and you can have anything in the freezer because it's just going to go bad, and eat anything in the food we have and everything. What they didn't say is you can raid the wine cellars too, but I did that. So what he became Party Central for all the newspaper and media people, the television people that came to New Orleans and needed needed foods and stuff. He's a good cook, cooks good gumbo, good jambalaya, the whole. So what gumbos and jambalayas are, they're just a bunch of things thrown together and thrown together well. So what Bob would do is he would make these big urns full of food, and everybody knew that at his place there was going to be a party that night and meals, and that went on for like 3 weeks. Every night, and so everybody knew who he was, still come and say hi to him and stuff like that. And that's making the best of a really bad situation. So next slide. What we have to do is just stick with it for as long as it takes to make things go the way they should, the way that is right and true and good. So how long will it take for the Starchild? Until we find someone with enough money, curiosity, and courage to join us in making it happen. When we find that person, every human's life will change in ways we can't really imagine right now, but I'm confident, I'm certain, that those changes will be right and true and good. Next slide. And for any of you that want to know more, this is the intervention and the e-books that you can get, but I'm not really trying to sell you anything. This is just going to be part of the slideshow. But that's it. So you can clap now.